Hello class, welcome to your lecture on gene regulation. This is module 15. We are nearing the end of the semester. We've only got one more module to go through after completing mod 15. Now mod 15 is based on content from chapter 11. So chapter 11 goes through three main sections. We are going to focus on the first and the last section. So we'll talk about the control of gene expression, and we'll also look at the genetic basis of cancer. That's a good way to understand gene control, is by relating it to, to a pathology that we are familiar with. All right, so to begin with the control of gene expression. So first of all, prokaryotes and eukaryotes do have some differences in the mechanisms that they use for gene control. So we'll first talk about the prokaryotic system, and then we'll move over to eukaryotes. So in prokaryotes, they utilize a structure called an operon. Here's the word for it. And this is actually a group of different genes that are associated with the production of a certain product. So everything is linked together to produce these products that are needed by prokaryotic organisms, such as bacteria. There are regulatory proteins that are part of the operon, and they bind to specific portions or specific sequences in the DNA. And like the name suggests, they have a role in helping to either turn on or turn off the expression or activity of an operon. So this is a very common operon. We're going to talk about two different operons, but we'll spend most of our time on this one. It is called the lac operon, and it is associated with the production of enzymes needed to process the sugar lactose. So the top part of the figure is when the operon is turned on, and the bottom part is when it's turned off. Now, one thing about the way that operons function is we've got two main types, and which to simply describe it, one type is always on, and then the other type is always off. That's the difference between the two operons we'll look at. Let's first focus again on the top part of this figure when the operon is off. So we're in the DNA. All of these sequences are in the DNA of the operon. So within the operon, we have the promoter, we have the operator, and then there are genes that are associated with the metabolism of lactose. There are three of them. So promoter, operator, and then the genes that are within the operon are all part of that operon. Right outside of the operon is the regulatory gene. So it is not in the operon, it is adjacent to it so that it can work really at a slightly different time, and you'll see what it does. So the regulatory gene is transcribed into its messenger RNA, and then it is translated into protein. And what it produces is a type of protein called the repressor. And so the regulatory gene encodes for the repressor. The job of the repressor is to bind the operon. And it is in between the promoter and the genes. So let's look at the structure of our repressor protein. It's got two little knobs that are sticking out of it, and those fit perfectly into the matching pockets within the operator. Now, what happens when the operator is bound by the repressor, it blocks RNA polymerase from binding, right? RNA polymerase, the polymerase binds to the promoter region. We learned about that in our last module. So that, again, I'll say it again, the RNA polymerase binds to the promoter region, which is part of the DNA that is within the operon. But in the case here, like we have up top, there's no lactose that's present in the system. So we don't need to transcribe and translate genes that are associated with lactose if it's not there. It would be a waste of materials to do that. So when lactose is absent, the repressor binds the operator, and that prevents RNA polymerase from binding to the promoter and then transcribing the operon. So I'm going to say it again. The regulatory gene encodes the repressor, and the repressor binds the operator, which blocks the RNA polymerase from being able to bind to the promoter. We're talking about RNA polymerase, not DNA polymerase. This is not DNA replication. This is protein synthesis. So the enzyme RNA polymerase, we can look at the name. It tells us that ASE means it's an enzyme that makes a polymer of RNA. And that's exactly what it does. It makes the messenger RNA that encodes that repressor. Okay, let's move to the bottom part of the figure. So now we're in the setting where there is lactose present. And lactose is shown with this little pink ball here. Well, it binds to the repressor and changes the shape of the repressor. So notice that those two knobs that are needed to interact to the operator, they're not present when lactose binds the repressor. It causes a conformational change in the repressor. And so now the repressor is inactive. It is not capable of binding to the operator. Consequently, RNA polymerase does correctly bind to the promoter region, and then it can move through and produce the messenger RNA that is associated with the three enzymes that are utilized for processing lactose. So lactose has to be present in the system in order for that to happen, in order for the operator to become on. There's no point in producing these lactose utilization proteins if there's no lactose in the environment. So this is the lac operon. It is usually turned off. It has to be turned on in the case of, let me start over again. It is turned on when lactose is present in the environment. Okay, so we've been talking about the lac operon. The proper name for this is that it is an inducible operon. This other example, the TRP, is for tryptophan. It is a repressible operon. So there's a fundamental difference in the kind of the baseline operation of these operons, whether or not it's constitutively on versus constitutively off. So when lactose binds to the repressor is inactivated, and therefore we do get transcription. So the way I like to think about it is that the presence of lactose induces the lac operon to turn on. In contrast with the trip operon, so this is for the amino acid tryptophan, it is inactive when there is no tryptophan present. When tryptophan is present, it binds to the repressor, which activates the repressor, allowing it to bind on to the operator, and then we would not have transcription of the trip operon because the RNA polymerase, which would bind to this green portion here, the promoter, it is blocked by the presence of this active repressor. So TRP has to be turned, the absence of tryptophan will make it so that the repressor is inactive here, and then it can't bind. All right, now let's kind of talk about a little bit about what differentiation is, what that word means. It just means it means that cells change and become specialized at what they do. So for example, a bone cell is different from a nerve cell, but we've got a lot of different types of white blood cells, and they're all different from one another, but they come from the same stem cell, the blood cells do. So differentiation is this process where a cell and tissue change so that they are able to become specialized for their job, for their environment. All right. Now, when chromosomes are packed down, right, that is not when the DNA is being replicated. A chromosome is very tightly wound DNA, but it also includes the histones, those small proteins that help to properly wrap the DNA, forming its correct structure. And it produces what is called a nucleosome. So that's DNA plus histones. So when we see that the DNA is packaged, this is an indication that there's not gene expression happening, because again, the DNA is so tightly wound down that the replication machinery is not accessible. So one way that scientists determine how active a certain gene is to look at the chromosomes that are associated with that gene. So an example of this happens with DNA packaging happens with a process that is called X chromosome inactivation. And this happens in females, mammals, and females have XX, right? They have two X chromosomes as their sex chromosomes, but only one of them is active. We're not utilizing both of those genes through gene expression. One of them is essentially turned off. It is inactivated. And there are changes that can happen to the DNA bases or even to the histones themselves. For example, certain chemical groups adding to them. And that can result in what is called epigenetic inheritance. This is a big area of research now, epigenetics, but it's a way to look at the effects of the environment and other factors on gene expression. There's a lot of research looking at epigenetic inheritance in different cancers. Okay, so let's familiarize ourselves with the structures that we've been talking about so far. So we're going to begin over here on the left. We'll look at the cartoon figure first. We've got our DNA double helix, right? So we've got our two strands of complementary anti-parallel DNA. And you can see these purple balls here are the histones, of which there are 8 of them per nucleosome. So the 8 histones bind in a specific way, and these are actually different types of proteins, which we don't need to concern ourselves with the types of histones. That's the topic for a molecular biology class. What we need to understand is the function of the histones, in that they help to package the DNA correctly. So a nucleosome contains the DNA that is wound around the 8 histones. That is what a nucleosome looks like. And we've got several of them here. We've got 1, here's 2, 3, 4, 5, we can see 6 nucleosomes in this example. This part of the figure here is an electron micrograph of the DNA that is organized in histones. And it's described as looking like beads on a string. And the beads are where the nucleosomes are. Then the string is just a little bit of DNA called a linker sequence that is in between each of the nucleosomes. Now, after the DNA packages itself in the form of the nucleosome, it then is wound again and again and again, down all the way down to the form of the chromosome. So we are looking at a duplicated chromosome here. We know it's a duplicated chromosome because it is existing as both. We've got the sister chromatid, and they are tied together by the centromere waistband. So in order, right, cell division would include breaking apart those sister chromatids and one going to each cell, each daughter cell. So this is an example of DNA packaging in eukaryotic organisms. And it's a relatively complex process, but it's absolutely necessary to get all of our DNA to fit in the nucleus of each of our cells. If the DNA were not packaged in the form of a chromosome, it would be too big to fit in the nucleus. But it also would be challenging for cell division to occur because it would be like a tangled mess of spaghetti, if you will. So that's DNA packaging. All right, the tortoiseshell cat is a great example to understand what X chromosome inactivation is. There is a reason why the tortoiseshell cats are female. There are a few examples of tortoiseshell male cats. However, in order for a male cat to have the appearance of a tortoiseshell, they need to have 2 X chromosomes. So they would be XXY. It can happen. But what's normally the process is we have our X chromosomes in a female. So there are 2 of them. So this is at the stage of the embryo. And on each of these X chromosomes one of them has the allele for orange fur, and the other X chromosome has the allele for black fur. And normally one of these would be silenced. But what happens in the tortoiseshell cat, how we get the appearance of 2 different fur colors, is there's inactivation of the different X chromosome. So in order to get the orange spots of fur, the X chromosome with the orange allele is what is being transcribed and translated to produce the orange color fur, where the black containing chromosome is inactive. The black fur comes from the other chromosome that has the allele for the black fur, whereas the orange allele is silenced here. So this is why the tortoiseshell cat characteristically has spots of black and orange fur on her. She's usually a she, and it's tied directly to the X chromosome inactivation process. It hasn't completed itself. It's still, right, we're still having expression from both X chromosomes in different regions of this animal. So just something to think about at this point, so we can get in the habit of taking the knowledge, taking what we're learning and applying it to a scenario. A nerve cell is very, very different than a skin cell. They have different structures and they certainly have different functions, but they all have the same genes in them. Every one of our cells has the full set of chromosomes in it, but when we're talking about a nerve cell, the chromosomes that are going to be expressed are going to be those associated with the nerve cell, not with a skin cell. So please make sure to understand that every cell has all of our chromosomes in them. However, they're not all going to be expressed at the same time, or else everything would be the same. So that's how cells are able to differentiate, and it happens based on the environment in which they're in during development. So there would be different enzymes available and active. All right, now we did learn in our last module that eukaryotic messenger RNA can be spliced. Let's take a look at how that happens. And it produces the ability to make more than one type of RNA, leading to different proteins coming from the same sequence. So this is properly called alternative RNA splicing because it gives us the option or possibility of producing different types of messenger RNA molecules, all from the same original transcript. Now, in humans, about 90% of the genes that are associated with producing protein do undergo alternative splicing. This is a mechanism to get more function out of the same material, right? Instead of having to have more DNA, where every segment of DNA is associated with one protein, for example, that would take up more space. So this is another example of evolution and this common theme of size, cell size. Okay, so let's look at an example of how we get two different transcripts coming from the same DNA. So our DNA is on top and it's in blue. We have 5 different exons. And recall that exons beginning with EX, they are expressed as protein. So we've got 5 exons and 4 introns that space out these exons. So what happens is the RNA transcript is produced. It is modified by adding the cap and the tail, and that helps allows the messenger RNA molecule to move out of the nucleus so that it can find a ribosome to complete the protein synthesis. Now, what happens with the splicing, notice that this transcript, this messenger RNA that is on the left side, it has the exons of genes 1, 2, 3, and 5. So what has happened is when splicing happened here, it took out the 4 gene. It spliced it away with the introns. Alternatively, on the right side, we have 1, 2, 4, 5. So now what's happened is the 3 gene exon has been removed. So we get 2 different messenger RNA molecules from the same transcript due to splicing. It is possible to get more than 2 messenger RNA transcripts from the same DNA sequence through the process of alternative splicing of the message. All right, let me go ahead and get this video playing. I'm gonna max out the volume.
As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus.
Okay, so that is RNA processing. Okay, this is actually a complex figure. It summarizes and shows you the many different levels where gene expression is regulated in eukaryotic organisms. So please pay attention to the number of mechanisms that are present here. And it's important that we understand what's happening with each of these. So first of all, DNA packaging, or the failure to unpackage, is a regulatory mechanism. If the DNA is in the form of a chromosome, it's not being expressed. So that right off the bat is one of the regulation mechanisms, DNA unpackaging. The DNA has to be unpackaged. It has to be unwound in the form of the chromosome so that the transcriptional enzymes and materials are able to get in there to use that DNA sequence as the template to produce messenger RNA. So that is the process of transcription. So if this process doesn't happen, that is a level of regulation. Splicing, which we've just talked about, alternative splicing, that's another way to regulate gene expression in that it can lead to the production of different gene products. Also, the presence of the cap and tail. If the cap and tail are not successfully added, this messenger RNA transcript doesn't have the ability to move out through the nuclear pore to complete the process of protein synthesis. So we've got splicing and addition of the cap and tail that would fall at the level of transcriptional regulation. That specific process of the message moving through the nuclear pore is another level of gene expression. All right. Messenger RNA is readily broken down. We have enzymes present in our body. They are commonly called RNases, RNase, enzyme that breaks down RNA. So we have these present all the time. In order for a message to be produced and for that protein to be produced, the message has to continually be produced from the DNA because it will be broken down by those enzymes. That's their job. And this is a level of gene control that prevents too much protein from being made when we don't need it. That would be a waste of cellular resources, energy, as well as time. So messenger RNA breakdown is another level of gene control in eukaryotic organisms. Now we're moving into the process of translation. So we've taken our messenger RNA and it has been translated into the primary structure of the protein. So that's literally the just like a pearl necklace. That's just the amino acid sequence. It's not functional though. It has to be folded to have its correct structure in order for that protein to be functional. So here's another level. The folding happens, different sequences are cut, and there are different modifications that can happen at the level of translation. So basically, if we don't get this protein through all four levels of folding, that would be through the primary, the secondary, the tertiary, and quaternary level of protein folding, well, we don't have the right structure. And if we don't have the right structure, then there is no function. And then also the protein itself is going to be broken down at some point back down into the amino acids that were utilized to build it. So that's another level of gene regulation in eukaryotic organisms. And in reality, all of this is happening. All right, so those are the different levels. So let's look at some examples of how a gene can be expressed. So this term, signal transduction, this is a pathway that results in the production of a protein. So we have two cells involved here. We have the signaling cell and then our target cell. The target cell has a receptor protein on its cell surface. The signaling cell produces the signaling molecule, which binds to its specific matched receptor. These are protein-protein interactions most of the time. So that means that there is going to be specificity based on the correct structural fit. So the signaling molecule binds the receptor. It does not come inside the cell. It stays on the cell surface. And this is true for any of our water-soluble proteins. They bind extracellular receptors. So once the signaling molecule binds its receptor protein, it sends a relay message down to the nucleus using a variety of proteins. And sometimes it's just, one or two. Sometimes there can be more members of the relay involved. But it's similar to what we all did when we were in grade school, when we ran relay races. So first, the message is perceived by the first relay protein, and then it passes it on to the next one, and then on to the next one, where it finally is associated with activating a transcription factor. And just like the name suggests, a transcription factor is a protein that is associated with transcription. We need to go in the nucleus if we're talking about transcription, because that's where the DNA is. So the transcription factor, once it's activated by the relay proteins, it moves through a nuclear pore into the nucleus, where it is then able to bind to the DNA at a specific place. And that initiates the production of that messenger RNA molecule, which then moves out through a nuclear pore and is translated to produce this protein. So this is a signal transduction pathway that results in the production of a protein. All right, let's take a look at this video.
Cell-to-cell signaling with molecules carrying signals from sending cells to receiving or target cells is a key mechanism in the coordination of cellular activities. In most cases, a signal molecule acts by binding to a receptor protein in the plasma membrane of the target cell and activating a series of changes, a signal transduction pathway, that triggers a specific response within the cell. A signal molecule binds to a specific receptor in the plasma membrane of the target cell. This activates a series of relay proteins, which in turn activates a transcription factor. The activated transcription factor attaches to the DNA and triggers transcription of a specific gene. The mRNA message is translated into a new protein, which changes the structure or function of the receiving cell.
Okay, so that is signal transduction, which results in the activation of a transcription factor, which interacts with the DNA and results in the production of the message. There are multiple different transcription factors, and different factors are associated with different cells and the production of different types of proteins. Now, prokaryotes have the equivalent of transcription factors too. All right, we're going to now move to the third section of chapter 11 and talk a little bit about the genetic basis of cancer. Now, cancer comes from mutations in genes that are associated with controlling cell division. Another way of stating that is that cancer is due to an overgrowth of cells that normally would be turned off through our normal cell control signals. We learned about the cell growth cycle earlier in the semester. So we're talking about mutations in that cellular machinery. So a mutation is capable of taking what is called a proto-oncogene and it converts it to an oncogene. And these are different. An oncogene is what is associated with uncontrolled cell growth. So how does a proto-oncogene become an oncogene? Now, proto-oncogenes are genes that are associated with controlling cell growth, cell division. There are certain viruses that are capable of converting proto-oncogenes and changing them to oncogenes, which will induce the cell to continue to divide indefinitely. So actually, when I went to graduate school, I graduated in 2003, and my major was immunology with a focus on virology. I learned at that time that there was one virus that was associated with changing proto-oncogenes into oncogenes, and that was the human papillomavirus that can lead to cervical cancer. We now know that there are several virus families that are capable of converting oncogenes or converting the proto-oncogene into the oncogene, including hepatitis C is another example, but there's quite a few of them. So the conversion, creation of an oncogene is one way that cancers develop. We also have other genes that are called tumor suppressor genes. And like the name suggests, they suppress the growth of tumors. And all a tumor is, it's a mass of cells. It doesn't necessarily mean it's cancerous or not. That would be the difference between something being benign versus non-benign. But when mutations happen in the various tumor suppressor genes, that makes them inactive, and then we lose that growth control mechanism. So an example of what happens with the proto-oncogene, and then we'll look at the example of a tumor suppressor gene. So we've got our DNA sequence that is associated with the production of a proto-oncogene, which functions normally. So we've got normal cell growth. If there's a mutation, and mutations can happen randomly, a virus is just one way to do this conversion, but there can be mutations that occur. But if there is a mutation in the right spot, it converts that proto-oncogene into a oncogene. You've heard the word oncology, which is the division of medicine that follows and treats cancer. So it's a cancer gene. And now we get too much growth. The cells will just keep growing because we've got too much of this oncogene protein being made now. Another example can happen where we have a proto-oncogene carrying multiple copies of a gene. And now we get our normal growth. We get too much of this growth, not normal growth. We get too much expression of growth factor, which then can lead to excessive cell growth. And then finally, if there's a mutation in a region associated with the control of cell growth, we again get too much of these growth proteins, leading to too much excess of that protein. So these are all three different examples of how an oncogene is converted from a proto-oncogene. All right, now we're looking at an example of tumor suppressor gene. And here we have our normal gene producing a protein that inhibits or arrests cell growth, and we get normal cells that divide normally. However, if there's a mutation in that tumor suppressor gene, then the protein is not going to be functional if it's produced at all. And now we don't have this control mechanism to turn off cell growth, to turn off cell division. So the cells grow and grow and grow and grow. They typically are bigger than normal cells. So that's one example of how they grow. They grow bigger, but then we have many of them that are growing in an uncontrolled manner. So this is what happens when tumor suppressor genes are non-functional due to a mutation. All right, so let's look at an example. We're going to apply this to colon cancer. And colon cancer is considered a preventable type of cancer. It is done using a colonoscopy, which is recommended for people usually about 45 years or older is when they should start to have this procedure done. The thing that, as you're going to see, there's plenty of opportunities to mediate the development of colon cancer. It's a slow growing cancer, so that gives time to do it. So we're looking in this example at the colon wall. And what happens first is an oncogene is active. And now we get increased cell division with these red looking cells, as you can see. So that is the first cellular change. This will lead to the growth of a polyp, which is a benign tumor. And this is what the colonoscopy looks for. And that procedure is done, by sticking a camera into a person's colon, and also attached to that camera are tools that can remove the polyp. So the way that we can mediate this disease is by doing colonoscopies and removing these polyps prior to them continuing the process of becoming a cancerous cell. So that's what the colonoscopy is used for diagnosis, but it's also part of the treatment in that many of these polyps are easily removed in that procedure. Next, we have a second example happening. So we've got another tumor suppressor gene that is inactivated. And this leads to the growth of the malignant tumor. So now we've got cancer. In stage 2, this is not malignant. Malignant means that there are cancerous cells and they have moved away from their original location. And we have invasion, you can see here, meaning that the polyp has grown and replicated, but it's moving away from its original site. So typically this is something that happens over decades, a decade or two. So colon cancer is considered 100% preventable with our very effective screening tools. So here's another example leading to a malignant cell. So we're looking here at a normal cell. We're looking at 4 chromosomes. First, we have 1 mutation, and we're starting to have a little bit of transformation towards the cancer phenotype and genotype. Then we have a second mutation, a third mutation, and a 4th mutation before the malignant cell is developed. Notice the malignant cell is shaped differently. They are. It's not looking necessarily bigger here, but many times these cells are bigger, but they don't look the same under the microscope. So when a person is, say, if a woman has breast cancer, part of that procedure in identifying if it has spread is the surgeon will look at the lymph nodes that are closest to the affected breast. And often while the patient is still on the operating table, they pull out some of these cells from the breast or the nearby lymph nodes and run over and look at them under a microscope, and then come back to the operating room to communicate that they found a malignant cell or not. So they're doing that by looking under the microscope. And If they identify a malignant cell in a lymph node, then that lymph node would be removed during the same surgery that is happening. So this is something that happens quickly, and it's just a procedure that takes a few minutes to take those cells, apply a little stain, and then look at them under the microscope. So a lot of value can come that can even prevent the person from having to have another surgery because they're doing this in real time. So we've learned about signal transduction, and this is, the way that the communication pathway that leads to the production of a certain protein or proteins from cells. And these specifically are proteins that are involved in the cell growth cycle. It turns out that, yes, many of the proto-oncogenes and tumor suppressor genes produce proteins that are part of this signal transduction pathway. These are the good genes. We want it to be a proto-oncogene. We want our tumor suppressor genes active so that they can inhibit cellular growth in the event of uncontrolled growth, which could lead to a tumor. All right, so we'll look at the signal transduction pathway associated with an oncogene protein. So this is normal. So we have a growth factor. That would be the signaling molecule now, since we're talking about signal transduction leading to an oncogene protein. So we have our growth factor. It binds its receptor on the target cell and sends the relay message through the relay proteins that are in the cytoplasm. It activates a transcription factor, which then moves into the nucleus, binds the DNA in a specific way, and that leads to the production of this protein that keeps everything happening properly. So we're turning off cell growth at the appropriate time. So this is the normal situation. Here's the abnormal situation. So now we have a problem in the growth factor, or there's none there. It could be improperly formed or absent. So we lose the opportunity to produce this protein. So instead, we don't have signal transduction. There's nothing, we're still sometimes able to activate transcription factors through other alternative pathways, but now we've got too much of this protein being produced, and it stimulates cell division, which could lead to the development of a tumor. So that is how the effect of a mutation in the growth factor could work. And it's going through, we're talking about a specific oncogene here called RAS, which is part of the cascade. We'll do the similar thing looking at the tumor suppressor proteins. So this is our normal situation, just what we've looked at. We get our growth inhibiting factor. Since we're talking about a tumor suppressor gene, it's going to be a growth inhibitor rather than a growth factor. It binds to its matched protein receptor, signaling the relay proteins, and which activate a gene called P53, which is a tumor suppressor gene. And then it moves into, once we get activation of this transcription factor, it moves into the nucleus where it's able to interact properly with the DNA, and we get the production of this growth inhibitory protein. So that would stop or limit cell division. When we have a mutant or faulty signal tumor suppressor gene, now we don't have the production of this normal P53 tumor suppressor gene because we don't have the proper protein to initiate the relay. So consequently, transcription and translation don't occur where we don't have the right materials. So there is no growth inhibiting protein produced. which means cell growth will go on uncontrolled because the tumor suppressor genes are not doing their job of producing the growth inhibiting proteins. So there are different factors that are associated with the risk of cancer, right? We've been talking about genetic factors, but there are also lifestyle factors that are associated with cancer. So presently, well, let's take COVID-19 out of the scenario, but cancer is the second leading cause of death in the United States, as well as in other industrialized nations. Heart disease is first. I believe presently, right now, that COVID-19 is the second leading cause of death in the United States, but typically it's cancer. And so we've got our genetic factors, but there are also environmental factors that are associated with the development of most cancers. And these are called carcinogens. Ogen means to cause, to generate carcinoma. So they are generating cancer. And that can be a chemical, it could be radiation. Those are the two categories of carcinogens, either a chemical or exposure to radiation. So by reducing our exposure to things like UV light, by wearing an appropriate sunblock when we're out in the sun, that is a way that even if a person has some genetic risk factors, they're able to hopefully mediate the development or hopefully reduce the chance that they would develop cancer. So now we're looking at, this is a little bit tricky to look at. There's a lot of good information here. So we're looking at different lifestyle factors that are able to reduce the risk of cancer. So we've got a bunch of risk factors. We've got alcohol, diet, being a female, if we're talking about something like ovarian cancer. Breast cancer certainly is more common in females, but it can occur in males. We've got genetic factors. For a male, we're talking about testicular cancer, for example. A person being overweight is associated with a risk for cancer. Pollution, radiation, smoking cigarettes, exposure to sun, sexually transmitted infectious diseases such as human papillomavirus. And then we've got other examples of viruses such as the hepatitis C virus and the HIV human immunodeficiency virus, are just to name a few. So those are the risk factors. What we're looking at on the x-axis are the numbers of new cancer diagnoses this is from 2015. So 10 times 1,000 is the way that you would read that scale. On the y-axis is the deaths that occurred in thousands in the same year. So the yellow means that there is no early screening available, where blue means there is. So for pancreatic cancer, there is not an early screening. That's why when people are diagnosed with pancreatic cancer, there's usually not much that's done. Notice how high the death rate is with pancreatic cancer. Look at lung disease. So our risk factors here are smoking and then pollution. And it does not have a mechanism for early screening either, and is associated with the highest number of new cases as well as the most deaths. Liver cancer is right over here. We've got breast cancer, which is more common than many other ones, and it is associated with deaths, but there is early screening available through mammogram. Mammograms aren't 100% effective though. About 15% of breast cancers are missed on a mammogram. And I don't mind sharing this with you. I was one of those people. I had a mammogram and it failed to detect actually a very large tumor that I had in my breast. So I feel that I would be remiss and not sharing with you that, that it's not a 100% effective screening tool. Women that have dense breasts are less likely to have the mammogram work for them because the tissue's just too dense to see through, if you will. So they would need another screening tool like an MRI. There are risk factors associated with it, including alcohol, of course, being female, genetic risk factors, and smoking, as well as obesity. But here's colorectal cancer, and it's common and it is associated with deaths. It is preventable through the colonoscopy and also by limiting exposure to these various risk factors. So there are things that we can do. And in a family where one person has a cancer, other members of that family may be at risk too of developing cancer due to the inheritance of a cancer risk gene. Presently, there are 46 cancer risk genes that are studied and utilized when assessing a person's risk for cancer. I imagine in 5 years, there's going to be more. They just haven't been identified yet. You've heard of the company called 23andMe that does DNA sequencing. Well, when they first started doing their sequencing, they were looking at a total of 23 or 24 different risk genes. But with good research, we're finding more. So I imagine that there will be others that are discovered as well. So family history of any type of cancer is a risk factor for the development of another type of cancer. It doesn't have to be the same cancer, because what is associated with cancer is the lack of cell growth control, right? So if we have a mutation in a growth control mechanism, that's a central mechanism that is needed for all of these types of cancers, not just for one type versus another. So what type of screenable cancer kills the most people? Here we go. It's colorectal cancer. And we've got a good 10, 15, 20 years to mediate this by getting in for a colonoscopy and removing those polyps so that the person hopefully does not develop cancer. There are plenty of people who are at risk of developing colorectal cancer that are maintained by the removal of polyps. And it's basically, I'm not going to say it's painless, because you got a big tube stuck up the rear end, but it certainly is a lot less devastating than cancer. All right, that finishes us up for module 15. You've got a few study slides back here. Definitely know all the different parts of the prokaryotic operon. It's important to understand that the repressor comes from the regulatory gene. RNA polymerase binds the promoter. The repressor binds the operator and blocks the transcription from happening when it's present. And then you've got your flow diagram as well. All right, that wraps us up for module 15. I hope everybody's doing well and that you're healthy and that your families are healthy. Take care. I'll see you back soon. Bye-bye.